This is a production of Cornell University.
Thanks, Anid, for that introduction. And yeah, you gave away my secret. I'm actually in Georgia right now. You probably can't tell by the cast iron wood stove behind me, but I am in Georgia. And yeah, thank you all for coming. Today, I'd like to present my dissertation work for my PhD. I've really focused my work on laboratory experiments looking at the role of microbial metabolism in the formation of mineral-associated organic matter. And I've put this picture here on the title slide to try to get us in the right frame of mind. This is looking down on a bee horizon that I've uncovered at Arnott Forest. And hopefully you can see there's a fungal hyphae stretching across the surface of this bee horizon. So this mineral-rich bee horizon. And my talk today is really going to focus at that interface, looking at organic matter decomposition and the role of microbial metabolism in bridging that interface between plant litter and stable organic matter. Let's see. So in the Martinez lab, we work at the molecular scale and ask mechanistic questions. And so I wanted to start off by just talking about soil organic matter from a molecular perspective and the way I think about it. So I've adapted this figure, but I typically think about plant litter input to the soil. It could come from various sources, detritus or root inputs. And the big distinction here is smaller compounds, such as low molecular weight monomers, DNA, RNA, lipids, and proteins, and the much, much larger polymers that make up plant material, such as cellulose, hemicellulose, and lignin. These inputs into the soil are then broken down by both biotic and abiotic mechanisms. So you might have depolymerization by exoenzymes or degradation from UV light or shrink swell processes that can then take these larger compounds and break them into their substituent components. Then you could have microbial biomass that transforms these compounds into non-living soil organic matter. So the main thing I want to point out is that there are two pathways, microbially derived and plant derived. So an abiotic and a biotic pathway for the formation of non-living soil organic matter. And then I really want to focus in on one portion of soil organic matter, which is mineral associated organic matter. And that's of importance to me because this fraction we know to be more stable in many situations, to have longer turnover times and longer radiocarbon dates. And also to have higher signatures of microbial transformation. So it's more microbial derived. And so I entered this field of research as a master's student at the University of Georgia. And I was fortunate enough to do some work looking at the impact of plant litter chemistry on decomposition and the formation of mineral associated organic matter. And what we found is higher quality litters, which we denoted as lower acid unhydrolyzable residues or lower hemicellulose and lignin content to higher protein content resulted in more microbial biomass and more mineral associated organic matter. So we could track the formation of mineral associated organic matter and collect observations that indicated that this was done because of increased carbon use efficiency. So not only was this potentially stable pool of organic matter being increased by this higher quality plant litter, but less CO2 was being emitted in the process. And so that really got me interested in this concept of carbon use efficiency and the microbial community as drivers of soil organic matter stabilization. For my PhD, I really wanted to zoom in to the molecular scale and from the microbial perspective. And so I really wanted to focus on just low molecular weight monomers and how they are transformed by different microbes in the soil environment and how that may drive the process of mineral associated organic matter formation. So the questions I came in with are, what is the role of substrate identity? There are all of these compounds in soil solution. What's the role of their identity on metabolism? Can we develop a framework that's applicable broadly across different lifestyles to predict monomer uptake rates and the efficiency of use of those monomers? How does phylogeny, so whether it's a fungus or a bacterial isolate, or the growth growth rate impact metabolism of these compounds? And then lastly, how do these metabolic processes impact the formation of mineral-associated organic matter? And so this was really interesting to me for many reasons. It's a very small scale mechanistic questions. They're very basic, foundational, and they're also increasingly being incorporated into models, these types of observations. So I was hopeful that I could collect some empirical data sets to help modelers move forward. So before we go further, we should define clearly what carbon use efficiency is and how I think about it. So this is my cartoon of a bacterial cell. It's in soil solution. It's surrounded by all of these compounds, potential substrates to create biomass from. Not all of them are readily available. They can't all be assimilated across the cellular envelope. So exoenzymes may be needed to break down these compounds into their monomer subunits. These compounds can then be uptaken or assimilated, either one at a time or sequentially or in groups. And that can facilitate growth or maintenance respiration, releasing CO2 as these organisms either survive or grow in the soil environment. And so we can calculate carbon use efficiency if we know how much carbon has been put into the biomass versus the total amount assimilated. So the amount in the biomass and the amount respired. And so that gives an efficiency, a proportion of that carbon that was maintained in the cell. So once you start thinking about these things at the cellular level, there are other interesting questions that pop up, such as how do we determine substrate preferences? Which compounds are assimilated in what order? And then are compounds used together? Is there co-utilization of substrates? Or do we see more classical dioxic patterns where one substrate is used after the other in a sequential fashion? And that's more of the classical paradigm of substrate preference utilization in microbiology. I want to define one other term, which is a finer point on carbon use efficiency, which is I'm calling substrate use efficiency. And that's in this mixed substrate environment, we could track the carbon from one compound with isotopes perhaps. And so this sugar, for example, even though multiple compounds are being assimilated, we can track the carbon from just that substrate into the biomass and into respiration and calculate a compound specific use efficiency rate, which here I'm calling SUE. So for my dissertation work, there were three main objectives of this work, and I'm going to share some of the work or some of the data from those projects. The first two objectives are focused on microbial metabolism, and we wanted to evaluate substrate energy content or the oxidation state of substrates as a potential predictor for microbial substrate preference and substrate use efficiency. The second objective is we wanted to test the impact of physiology on carbon use efficiency and any relationship we observe between energy content and substrate use efficiency. And then the third objective is separate and is really focused on the formation of mineral-associated organic matter. And we wanted, for this talk, we wanted to assess the functional groups involved in formation of MAOM from microbial necromass. So to carry this out in a practical sense, I employed a model systems approach. We chose a field site at Arnott Forest, a site that's also been researched by many others, including the Goodale Lab. We chose a hemlock hardwood stand. Oh, so I forgot to say this is right near Newfield outside of Ithaca. And we chose a hemlock hardwood stand to study. This is a young inceptisol, well-drained, and we sampled the OA and B horizons to get material for further experimentation. I then needed to create a soil solution analog to culture microbes in. So I made a water extract, a tea, if you will, of this OA horizon. And so it's got this orangish tint. It's pretty acidic, but the idea here is that it's an analog for a rainfall event where you have aerated water percolating downward and being utilized by the microbial community. Then we needed to characterize this tea we made. So we hit it with a suite of analytical approaches to try to understand what carbon substrates were there and in what concentrations they were available to the community. We then created an isolate library where we chose 20 isolates, both fungal and bacterial, isolated from this site. We also isolated them on the soil extract. So the selection pressure was that we chose organisms that would likely respond to a rainfall event, to the percolation of this carbon-rich solution downward. We then used 16S and ITS amplicon sequencing to better identify them. We characterized their growth rates. So the first chapter I'm going to talk about, I took one of these organisms and grew it in the soil extract, and we quantified substrate uptake, growth, carbon use efficiency during growth in that soil extract. We then used the data from that experiment to create a defined media. For practical reasons, extracting enough of this soil tea every time, it just wasn't feasible. So we created a realistic defined media with many carbon substrates at low concentration. And then we used that to conduct an experiment looking at substrate preferences, substrate use efficiency, and carbon use efficiency across a growth rate gradient using isolates from our library. And then lastly, we brought in a mineral component, isolating clays from the site, from the B horizon, and also synthesizing a model iron oxide goethite for use in further sorption experiments. And so that's the third chapter of my dissertation, looking at source effects on the retention of microbial necromess. So before I go further, I wanted to just give the hypotheses for the first two experiments. So again, we're tracking the fate of low molecular weight organic substrates in solution. And the first hypothesis that we wanted to investigate, or the first hypothesis I had that we hoped to collect observations for against, was that higher compounds with higher nominal oxidation state of carbon, more oxidized compounds, would be used less efficiently by various isolates. And this was resulting from literature in the physiology, in the field of microbial physiology and soil science. So this plot shows substrate use efficiency and then nominal oxidation state of carbon on the x-axis, going from most reduced, such as methane, to most oxidized, like CO2. And research from the physiology literature, Gommers et al. Reviews it nicely, showed that for individual carbon substrates used alone, there's a clear trend where more oxidized compounds are used less efficiently. And there's a good thermodynamic underpinning to why this is the case. But the interesting thing is when we spike soils with these different compounds, we see similar trends. So that's the other colors overlaying here. But it's less clear if that's because of a universal framework within the metabolism of all microbes, or because of which microbes are using these compounds in a mixed community. And then the second hypothesis that we wanted to look at was how growth rate of different isolates impacts overall carbon use efficiency and any relationship with the use of specific substrates. And so this is an observation that has already been incorporated into global models, actually, that faster growing organisms tend to have lower carbon use efficiencies, and slower growing organisms, have higher carbon use efficiencies. And that's based on quite a bit of culture work, but there's less work been done at concentrations that are realistic for soil solution. So we wanted to collect data to confirm or disprove that. So to begin with, the first thing we did was characterize this soil solution. I took some basic measurements of pH and electrical conductivity. It's a very acidic solution, which isn't surprising because we're under a hemlock forest cover. I did find more carbon than I expected, roughly 183 milligrams of carbon per liter. We can see that there's some ammonium. I found no nitrate, and that most of the nitrogen was in the form of amino acids or other organic nitrogen. And that with colorimetric methods, I could see quite a bit of the carbon was in the form of reducing sugars. So that's very, crude bulk observations of the solution. We then could use fancier exometabolomic methods, proton NMR, and liquid chromatography to quantify specific low molecular weight substrates. So here we have concentration on the y-axis, and then roughly, I believe, 30 compounds that we quantified on the x. And you can see that we saw a large gradient in concentration, ranging from 0.4 to 236 micromolar. So the main takeaway here is that there are many substrates, and they are at very diverse concentrations in this soil solution. So what we could quantify only explained 20% of carbon and 40% of nitrogen, but that's a pretty sizable fraction for such a small number of compounds. We identified another 150 compounds that we couldn't quantify, but we know are in solution. So now I want to show some results from the first chapter. This was an ecophysiological study of one of the isolates, Paraburkholderia solitsugae, on soil extract. And this was a nice side collaboration during my PhD with Rolly, Wilhelm in the Buckley lab, and several other members in the Buckley lab. And we were able to take one of these isolates, further characterize it, and actually name it. So this is data on that organism growing in soil extract. It's a gram-negative Paraburkholderia species. So we grew the isolate to log phase overnight, and then we used that to inoculate culture flask with 12 replicates. Then with time, we could sample those flasks and measure carbon, nitrogen, biomass, and protein, and then filter to conduct our exometabolomic measurements to see what was changing in extracellular solution. So here is the growth curve. To orient you, on the y-axis, we have optical density at 600 nanometers, which also correlates to biomass over here on this axis and protein content. And then on the x-axis, we have time in hours. I've fit the growth curve with a black line here, but the actual sampling points are in red. So we sampled 4 points along the curve, trying to capture log phase. And I've included the starter flask here just to try to prove to you that my modeled fit for stationary phase is realistic. I want to go back real quick and just note that we missed quite a bit early on, even though we did capture a portion of the curve, but you'll see that come into play later when we look at specific substrates. So I don't want to spend too much time on this, but I just wanted to show that we have bulk measurements of carbon and nitrogen dynamics in solution, that we could take these, and with time we could follow movement of carbon outside of extracellular solution into the cell and into respired CO2, for instance, and we could track kind of bulk species such as reducing sugars, right? Same with nitrogen, we can track ammonium uptake here in the orange color, or amino acids in the pink. And from that, we were able to see that just over a quarter of carbon available in solution was assimilated by this microbe. About 18% of that initial carbon was lost as CO2. We could explain a large fraction, roughly 30%, with reducing sugars, and an even larger fraction with amino acids. With this data, we could also calculate carbon use efficiency. And so here I've got CUE on the y-axis and time on the x-axis, going up to 24 hours. And what I want to show here is that this fit is for cumulative CUE, and we can see that increases with growth, plateauing at about 0.4, or 40% of carbon assimilated was maintained in biomass. And if we look at instantaneous carbon use efficiency, we can see that that's much higher at these during this growth phase. So, you know, CUE is very low early on. It rapidly increases, right? And then slows down again as growth slows down. So this value of 0.4 is right in the middle of what you'd expect in the literature. It also happens to be the ballpark number used in many models. And it also fits some emerging relationships between growth rate and carbon use efficiency. So the point there is that our measurements seem to be working and producing realistic values. Okay, moving from here, I want to show some of the exometabolomic data. This is just one example for citrate. So citrate was in this soil extract. We can track its concentration changes with time, and then we can fit a sigmoidal curve to the uptake of that compound. And so from that curve fit, we can extract parameters of interest, such as the amplitude, how much was assimilated, the T50 or the inflection point of assimilation, the width, or which relates to how fast or slow the compound is used. And then what I did is I took that width and I expanded it to show a 90% usage window. So we can really look at compounds at their 10% used and 90% used points. And so I'm showing these fits because in the future plots, I'm going to just show the T50 and the 90% usage window. So we can look at when the substrates are used along the growth curve. So here is this figure. This is for the growth curve of Paraburkholderia, solitsugae in soil extract. So again, we have time on the x-axis, biomass on the y. The black line is showing biomass growth, accumulation. And you can see that we see distinct clusters of substrate use. So I missed quite a few early on, which I've called type 1 here. And I've just counted them over here. But each of these is a T50, so a midpoint of usage, and the bars on the x-axis show the usage window. So the main takeaways from this experiment was that our quantified compounds explained about 3/4 of carbon and nitrogen removal from the media. So while we only quantified a certain amount, it explained a large portion of what was actually being metabolized. And we saw distinct clusters of substrate depletion, at least 3, maybe more. And this distinct clustering, in my mind, implies multi-oxic growth. So we have different groups of simultaneous substrate usage. And the one other thing I want to point out, which maybe if you're good at squinting, you can see in this figure, but I'll show a regression, is we saw no clear trend with nominal oxidation state and substrate preference. So if I plot T50 on the y-axis versus nominal oxidation state on the x, we see no clear trend. And so the colored points are quantified substrates, and then I've also overlain untargeted compounds. So compounds we can't quantify, but I can assign them or calculate a nominal oxidation state. So we saw no support for that hypothesis in this study. So from there, we had to make a decision to make defined media. So we looked at what substrates we could quantify, and we really at that point wanted to remove concentration as a confounding variable. So we chose 25 micromolar as a reasonable concentration to make a defined media with. And we moved forward with creating that media with 34 total carbon sources, 3 sugars, 21 amino acids, and 10 organic acids. Again, they're all at 25 micromolar. We supplemented with ammonium because that was present in soil extract as well as phosphate. And then we also supplemented with Wolff's trace mineral solution in vitamin mix. We bumped the pH up a little here, but it's still at an acidic pH of pH 4. And we use this for our further experimentation. So for this work, we really wanted to ask questions again about substrate preferences, substrate use efficiency, and carbon use efficiency, but across multiple different isolates. So not just the Paraburkholderia solitsugae. So we chose 3 species with different growth rates, and I've just put the name of their closest relative based on amplicon sequencing. And so we have 2 gram negatives, a Ralstonia, and then the Paraburkholderia I mentioned earlier, and then a fungus, Penicillium spinulosum. And this represents a growth rate gradient. So the fungus is much slower growing than the 2 gram negative bacteria. We then chose 5 substrates in particular to add as labeled, as isotope labeled substrates, so we could track the carbon and nitrogen from those substrates specifically. And here we had 2 amino acids, valine and glycine, a sugar, glucose, and then 2 organic acids, acetate and formate. And then from a practical perspective, the way this experiment was conducted is we grew up these isolates into log phase and then used those to inoculate a culture flask containing the media with all 34 substrates, but just one isotope labeled substrate for each treatment. Then we could take gas measurements to monitor CO2 production and labeled CO2 production, collect biomass with time, monitor substrate uptake. And for each of these experiments, we did at least 4 time points sampled. So there's a lot of data here, but I'm just going to point out a couple quick takeaways to orient you. I'm showing the microbes on the bottom with decreasing growth rate. For each of these figures, biomass is on the y-axis and time is on the x-axis. You'll notice that the x-axis gets larger as you move to the right. That's because they're growing slower, so the growth curve takes more time to capture. You'll also notice that the y-axis increases as well as you move to the right. So with decreasing growth rate, there was more biomass production. And I just want to point out that all of these microbes assimilated all of the substrates that we put in the media, which perhaps isn't surprising, but all sugars, amino acids, and organic acids that we put were assimilated, and we could monitor that with time. Again, looking at the T50, so the inflection point of uptake in the usage window. And so again, it's hard to squint and bring out a pattern with oxidation state, so we can do a regression. So here I'm looking at the inflection point, the T50, of each substrate as a function of the oxidation state of the carbon in that substrate. And what comes out of that is that we see indications of a trend supporting our hypothesis for the 2 bacterial species. So we see that the more oxidized compounds are used earlier on during growth. And we see less of a trend for the fungal species. And so I'm in the process right now of going back through and making sure that I'm confident in all of these fits, because we've done a lot of sigmoidal fitting here. But this seems to be the story that's coming out here, is that we see a trend with the bacterial isolates, but not with the fungal isolate. So then we can look at the respiration data. So again, we have 3 figures, and I'm showing enriched 13C CO2 on the Y and time on the X. Each of the 5 labeled substrates I mentioned earlier have a different pastel color here. And again, the main takeaway here is that I can capture kinetics of respiration of these compounds. And then to calculate substrate use efficiency, I can take the final value, the cumulative respired 13C CO2, and use that in my formula. So if I know how much of that isotope is in the biomass, and I know how much was cumulatively respired, I can calculate SUE. So what I did is at the end of these curves, I can take this final value along with a measurement of the biomass to measure SUE. So here are those final plots. Again, we have nominal oxidation state of carbon on the X and substrate use efficiency on the Y. The first thing I want to point out here is that decreasing growth rate did result in higher carbon use efficiency. So in this more realistic soil solution environment media that we've chosen, we see that the fungus grows much more efficiently than the 2 bacterial species, and we see a trend with growth rate. The other thing I want to point out is that there are not clear trends with nominal oxidation state of carbon. So if we look just at the Paraburkholderia species, we can see trends similar to what we see in the literature, where the more oxidized formate is used much less efficiently. But that's less clear for the Ralstonia or the Penicillium species. For all cases, formate was used much less efficiently than any of the other substrates. And we see some indication by inference. You can see that there are other substrates in the media that must be used less efficiently than these 3 for Ralstonia, because the average is much lower. The same with Paraburkholderia as well. But it's less clear with the Penicillium, where it looks like the average matches much more closely with some of the substrates we chose to monitor. Okay, so I'm going to move on now to the final portion, which is focused on adsorption. And so the questions we wanted to ask here is what functional groups are predominantly involved in necromass sorption? Do these differ with phylogeny? And then what is the stability of that mineral-associated organic matter after it is formed? And so to do this work, we use an instrument that's the workhorse of our lab, the ATR-FTIR. And this instrument allows us to collect interfacial observations or collect FTIR data at the interface of the mineral and solution. So we can place a mineral film on this diamond, stabilize it, and then flow a solution over it, monitor sorption in real time. So here's an example. This is just one of the many experiments I've run showing sorption of bacterial necromass. And so, with time in red, you see sorption. So what starts off as, no vibrations, quickly we see peaks show up, and we can assign those peaks to different functional groups. And then I haven't shown it here, but with time, we can also watch desorption as these spectra drop back down if we change the environment or change the solution flowing through the instrument. So for the sake of the talk today and for time, I'm going to be very simplistic with this and just point out two portions of microbial necromass that we can focus on because they look quite different, and that is protein. So protein and DNA, and here I've shown two spectra from an old lab mate of mine, Mike Schmidt. And so for the protein spectra, we can see that it's dominated by amide 1 and amide 2 bands, which we also see in our necromass that's sorbing. And then DNA, which also has vibrations in this region, but has much higher relative vibrations that we can assign to PO or CC backbone vibrations in the DNA molecule. And I'm just going to get you to focus on those two end members for the rest of the talk. So for these experiments, I took necromass from these different microbes, and I adsorbed them to a goethite thin film. So first we stabilized the film with background KCl, an electrolyte that's common in soil solution. And then we sorb the necromass by flowing that repeatedly over the mineral. And so that necromass has been sonicated, the cell has been broken open, and it's sterile. So we conduct sorption, and then we change the solution to induce desorption. First, we just remove the necromass, just have a background electrolyte. Then we increase the ionic strength tenfold. And then we add an organic ligand, oxalate. And we chose that because it has commonly been shown to induce desorption. And so we wanted to see if we could use that to kick off some of this sorbed necromass. So again, here I've shown three figures for the three isolates I've chosen to work with. For now, these red spectra are just the necromass themselves. And I don't want to get too caught up on individual peaks, but the main thing I want to point out is that the microbial bacterial necromass is much more similar. It's dominated by a protein signature, so the amide 1 and amide 2 peaks, and that the fungal necromass has a much decreased relative protein signature and looks a lot more like DNA. So it has a higher relative COC, CC, and PO vibrations. And so those are over in this lower region, right, versus the amide 1 and amide 2, which are clear in these two bacterial isolates. So then we can look at equilibrium, at sorption on the goethite thin film. And the main takeaways here that I want to get at is that we see selective preservation. So the spectra are different, right? We don't see all portions of necromass equally sorbed on this goethite mineral. We see increased relative COC, CC, and PO vibrations, which are all DNA associated or can be upon sorption. So then ongoing work that I'm involved with right now is looking at the kinetics of sorption and the stability. And so we can break this spectra up into regions of interest. And for the sake of this talk, I'm going to mention amide 1, amide 2, which are both related to protein. And then this region that's more associated with DNA down here. And we can monitor those by integrating those regions. So we can monitor those with time during sorption, when we change to just our background electrolyte, when we increase the ionic strength, and then when we add an organic ligand. And so we can see sorption and then desorption using this approach. And so again, if we look at our 3 isolates, the main thing I want to point out here is that we see sorption for all of them, and then we see desorption with our phases. And we see distinct kinetic patterns for both sorption and desorption. And so the work that I'm currently doing to finish up this chapter is modeling the kinetics of these sorption and desorption events and comparing them between the 3 isolates. And let's see. Yeah, so I think overall, to wrap up, I hope I've shown or convinced you that exometabolomic methods are a really important way to better understand microbial physiology at realistic substrate concentrations. So because of these low detection limits, we can grow and monitor these microbes at very low substrate concentrations. We saw evidence that faster growing microbes tend to metabolize carbon less efficiently, so they had a lower carbon use efficiency. We saw some indications that more oxidized compounds were used earlier during growth, at least for the bacteria, the 2 bacteria we investigated. So higher nominal oxidation state of carbon resulted in lower T50 or earlier uptake. We saw no clear trend between nominal oxidation state of carbon and substrate use efficiency. And then lastly, I have some preliminary data showing that preferential adsorption of DNA-associated functional groups from necromass from all isolates. And I'm currently working to compare the kinetics of those processes. And then, of course, there are always caveats. The uptake relationship that I did see was only clear at equimolar substrate levels, right? If you think back to the first chapter, we didn't see it in the soil extract. So if we were to incorporate that into any modeling efforts, substrate concentration would need to be a factor there. I also want to caveat again, I'm only talking about aerobic respiration here. Things may be very different in other metabolic environments. And then lastly, I've done this work using individual isolates to ask questions about basic microbial metabolism, but no microbe is an island. And so things may be different once you're operating in a community. So just wanted to throw those caveats out there. None of this work would be possible without lots of help from those in my lab, including Mike, Ariel, and Behruz, the Buckley lab, who have graciously let me use their lab space and put up with me for all these years. I'd like to especially thank Dr. Roland Wilhelm for his mentorship, and Dr. Lumia Aristilde and Dr. Annalise Klein for their help with exometabolomic measurements. And so with that, I'd like to thank everyone for their attention and open the floor for any question. Shoot, Dominic.
Hi, Taylor. Thank you. That was a wonderful presentation. And as a modeler, I really appreciated you saying early on that you wanted to produce stuff that's useful to modelers. And I think you've definitely done that. It's fascinating to me to think about how I can put some of this into models. I'm wondering a little bit about the carbon use efficiency. So you didn't find strong evidence of oxidation state being a good predictor. And from the modeling community, people typically expect it to be driven far more by the C to N ratio, because growth has a stoichiometric demand for carbon use efficiency has to match the stoichiometry of growth. Did you think about trying to correlate your data with the CN ratio of the substrates and whether that's a better fit? And I said, for example, with formate, it's also got a extremely high and infinite C to N ratio. So we would expect on that basis to see the low CUE that you found. I wonder if C to N could be mapped against that, or whether you did that.
Yeah, thank you so much for that question. So just to reiterate, the question was if I'd looked at C to N ratio as a possible predictor. And yeah, I mean, I think coming from the larger scale work, like my master's work was found that AUR to N, which is just a different version of C to N ratio, was very useful as a predictor at that scale. But at these scales, I saw no relationship with that either. And then the other issue is some of these compounds, like you mentioned, have no nitrogen at all. So how do you, I took subsets of the amino acids and tried to use that as a predictor with no use, or with no clear regression either. Thanks for that question. I hope.
Yeah, thank you. Yeah.
Hi, this is totally outside my area, so this may be not a helpful question at all, but it looked like with some of your comparisons, you were using the inflection point, which of course is harder to quantify than if you moved a little further along in the time course. So instead of using a 50th percentile, use some higher percentile. I'm just curious about, I guess it's sort of a methodology question of why you choose that for the further fitting.
Let's see, let's think through this. Trying to find the slide.
And again, this is outside my area, so this,
No, that's a good question. I guess my thinking, like just off the bat, thinking about that would be that because of the way the sigmoid is fit, if I choose, let's say 75 or 25, oh, I see what you're saying. I guess to me, I was just thinking about centering it, right? To think about the midpoint of use. And then, depending on the shape of that sigmoid, you'll have a wider or smaller uptake window. But you were saying like use like the 25th percent?
Yeah, like it's just a question. It seems like the highest uncertainty is at that 50th percentile. So you would have more, seems like you'd have more precision if you used, say, a higher percentile.
Okay. I can certainly try that. That's easy, just, calculation and replot. So I'll think about that. Thank you.
I was wondering, Cyle, thank you very much for this wonderful presentation. It looks amazing, all the work you did. I wanted to go back to the NOSC question and how well the NOSC, your compounds approximate the average of that you found in your concoction. And for the fungus, you didn't find. It sort of aligned with the average, so not that it characterizes necessarily what's in that concoction, but what are you missing in the bacteria that is not in your model substances here? Yeah, exactly. So what other compound classes that you think could balance the budget here to get you to the average?
Yeah, so that's a really good question. Thank you. So the good thing here is this is defined media, so I know exactly what is available. There's only 34 options. So in the case of the two bacteria, it could be that they're using several of those amino acids, or probably organic acids, less efficiently, right? And that would be it. Just, from a practical perspective, I could only probe 5, right? But if we could probe them all, then we could ensure, right? And I would expect a lot of the others to be below this curve, right? So I guess short answer is I would guess organic acids, but I can't say for sure without probing that. And then the fungus, like you mentioned, is more interesting because it almost insinuates that most of the substrates might be used with the same use efficiency, right? And maybe this formate is just an outlier. And so I'm trying to think through, it kind of really gets at a core question is, are these substrates being used at vastly different use efficiencies or being used at the same time divergently in metabolism? Or are they all being used with similar use efficiencies?
And so it seems that your organisms are doing very different things, but the compounds have a less of an effect. Well, maybe there's a little bit of a slant in all of your data, but but maybe less than you expected. So does that mean that organism is more important, or organism class more important than compounds for this whole growth question?
Well, yeah, I think clearly the metabolism of the organism is the most important thing, right? That's going to define how these substrates are used. And what I guess I was hopeful to find is if we could kind of forget about that, right? And find a broader framework, because that would be great for modeling, right? You wouldn't have to measure the specific metabolism of each of these organisms to predict, right? You could have just a large framework. And so I don't know if I don't think I have compelling evidence that we can use this one parameter, right, and solve the problem and predict uptake. So
yeah, but it's always one organism, right? I mean, as you said in your caveat, that you're not looking at a community of organisms.
Oh, so that's a good point. In a community, right, we may see a shift where, let's say I'm this middle organism, Paraburkholderia, solitsugae, maybe I can't get at the glycine as fast as someone else in the community. So how efficiently I use that may be less of a factor, right? Because there's competition there.
Yeah.
Or I might have decided to focus on one compound because that's my niche, right? There's lots of ways we could think about how that might play out.
Great, thank you. Wonderful.
Thank you.
Oh,
I think we have another question.
I see.
Yeah, oh yeah, I turn on my video. So thank you, Taylor. That was a very, very interesting talk and good seeing you.
Good to see you too.
I have a question concerning also the carbon use efficiency and how it was measured experimentally, because I might not have caught everything you were talking about. You are measuring the optical density in your solution and you take that as a proxy for the growing microorganisms. Is that correct?
So let's see if I can go back. So very good question. I know I went quickly through this. So I measured optical density, which correlates with biomass and with protein. But I actually physically measured biomass with this, if you can see my arrow, with this filtration step. So the assumption here is that everything greater than 0.2 microns is my cell. We're going to say that's biomass. And so I could physically weigh that, mass that, as well as measure the amount of carbon by difference. So I could take a solution over to the Shimadzu unfiltered, and then a solution filtered, and by difference know the carbon in the biomass.
So, okay. So you used the filtration of larger than the size of a bacteria or fungal to say that's my living biomass, to separate it from dead material.
Yeah, so that's a good point, is that it's not perfect. There are maybe some dead cells in there, right? But that the assumption is once the cell breaks open, that a good portion of that should go through the filter.
Yeah. No, it's very interesting because I know it's difficult to measure. So it's relatively easy to model it, but to get hard data on it, we are having difficulties. So I'll probably come back to you and ask more questions.
Yeah, I know it's very hard to measure in an actual soil. And I almost laughed when this one organism ended up being 0.4, which I think is the default modeling number.
Okay.
Thank you for your question.
Thanks for answering.
Okay.
Hey, Dom.
Yeah, am I allowed another question? Because I didn't see other hands up.
Please.
Oh, yes, sure.
So I was wondering about how we would translate this. Maybe you haven't thought about it, so maybe it's an unfair question. How do we translate this to what happens in a soil? Because we often find that use efficiencies are very much higher in an aqueous broth because you don't have to forage or compete with other organisms or compete with uptake against minerals and all that sort of thing. And I think we typically see, I don't think you ever see CUEs in the sort of 75–80% in a soil environment, whereas you do see them during the growth phase in an aqueous slurry, which is a much easier environment for an organism to live in. So I'm wondering how would it be possible to take what you've done here, and what lessons could we actually translate into the soil environment, and which ones are very specific to an aqueous solution?
Yeah, I totally agree with you that in a real soil, things will change dramatically. And I think my basic goal was to provide some parameters, because I see these estimates starting to be, or these types of numbers like uptake kinetic values being put into models, and there's very little data to parameterize that. So if nothing else, I hope to provide like bounds on what uptake rates might be in solution. But you're right, the carbon use efficiency of one of these isolates might change drastically if I put a mineral in the flask, right? But the problem is it's just very hard for me, at least with the methods I had available, to measure CUE in that situation, right? So
yeah, no, I agree. It's a really difficult problem to tackle. It was so much easier to work in these simplified environments. I'm just wondering, like as a modeler, you know, how much can you take from these environments and assume that things look anything like that in a soil? And maybe that's a stretch.
I have to, I mean, we are totally over time, but I just want to say that, Taylor, you did have some experience trying to do that experiment that Dominic was talking about, right? Basically putting the mineral in the flask. What happened there?
Yeah, so that's a fun, I could give a whole nother talk on failed PhD experiments, but I started off at one point trying to add minerals into the mix, right? And the problem is everything died instantly. And so I spent a couple months on, it was fun actually, on side projects trying to understand if the microbes were dying because my goethite was sorbing all the phosphate. And so then we have a nutrient limitation issue, or if it was directly killing the microbes. And there's a whole interesting side literature on the needle morphology of goethite and how it may pierce bacterial cells. If you're looking for like a good Sunday afternoon read, I encourage that. But yeah, it's hard, I guess, is where I'll end with that.
Yeah. All right. So yeah, so it's 1:20. Oh, okay, good. So I just have a question. I don't see any more hands. So I think we can close shop for today, right?
Okay, and if anyone has other questions, you can email me at any time.
This has been a production of Cornell University, on the web at cornell.edu.